Hi, everyone. Welcome to Microbial Minutes for July 23rd, 2018. This is the American Society for Microbiology's every few weekly update on what's hot in the microbial sciences. I'm Julie Wolf of the American Society for Microbiology, and we're going to talk about several different things that have been going on in the last 3 weeks since our past session. First, we're going to start off with some microbiome news about a new couple of databases, and then we're going to move into Candida persistence and drug resistance. Next we will move into the results of a recent HIV vaccine clinical trial. Then we'll move into some antibiotic updates. Who is prescribing antibiotics and who is making them? And finally, we'll end with a new treatment for smallpox, which might not seem necessary, but we'll discuss that as well. Let's dive right in with our microbiome news. We're going to talk about two different papers that I thought deserved coverage. The first is from Nature Communications, titled Gut Microbiota Associations with Common Diseases and Prescription Medications in a Population-Based Cohort. And the take-home message from both of these is really that new databases link microbiome compositions to disease. Microbiome is a really hot topic. Everyone wants to be able to understand what those different bacterial communities are, the different microbial communities are, how those ratios affect human health. And we've discussed several different types of diseases that are associated with an altered microbiome state. That may mean something like different disease ratios, it may mean something like different bacterial ratios, but it also may mean something such as a new member of that consortium. And so rather than look at this on simply a one-on-one disease versus health state, this first research group, wanted to investigate it on a larger scale, looking at human populations and the many different diseases that are associated within a population. And so what they did was to take the data from the Twins UK cohort. This is 2,700 members, and it's been a long-term study, so there's a lot of different data available about the members of this cohort. It's been ongoing, I believe, since the late 80s, so quite a lot of different characteristics of these different twins, of the cohort members, things about their state of health, what medications that they're on, what genetic and or infectious diseases they may have. And so the researchers looked at their microbiota, looked at their gut microbiota, and investigated these for associations with one of 38 common diseases and one of 51 given medication. And so what they were able to do is to look for given associations of gut microbiota markers for each disease. So they were looking through the literature saying what different diseases are associated with certain types of microbiota constituents. And then they looked at the individuals of their cohort and they looked at who was sick and had a given microbiota marker, who was sick but did not have that marker, and who was sick or who was not sick but had those markers present in their fecal samples. And from this, they were able to draw conclusions and associations about different microbiome members, different bacteria that are good or bad and associated with different outcomes for human disease. So if we look at this association graph that they published, you can see a number of different diseases that are seen along the x-axis here. And the red or blue indicate whether something was associated with a negative health effect in the red or a positive health effect in the blue. And although the different classes of their bacteria that they looked at largely grouped into blue on the bottom and red on the top, you can see that there are certain blue spots within the red of the top and different red or negative health effects associated with these mostly beneficial bacteria. And they looked at a number of different types of diseases. They didn't only look at gastrointestinal disease. They looked at things such as hearing loss. They looked at COPD, which afflicts the lungs, as well as a number of GI afflictions such as constipation, colitis, IBS, things that one would expect would have associations with different gut microbiota markers. They also made this same association with the drug microbiome associations by looking at who within their cohort was using particular drugs and whether there were markers that were able to be associated with that. However, this is just one piece of a large puzzle. So lots of different research groups are trying to make these associations between a healthy and perhaps a disturbed microbiome. And it's hard to assemble all of the literature into one place. And that's where the second paper comes into play. This is something that was published a few weeks ago in BMC Microbiology, titled Dysbiome Database, Linking the Microbiome to Disease. Excuse me. And what these researchers hope to do is to combine the previous research with all genomic, protein, diversity, and other databases that are available about microbiome and the potential links to disease into a single database. And so by visiting this website here, dysbiome.ugent.be, you arrive to their homepage where you can type in any type of disease or organism that you want to investigate. And this will pull from 190 different diseases, 800 different organisms, 25 different detection methods, and 50 different sample types. So as you can see, I looked up celiac disease and it brought a whole number of different organisms that are associated with different states of the disease. It indicates the method by which this was associated, whether it's fluorescent in situ hybridization or 16S ribosomal RNA gene sequencing. A lot of these different techniques are listed, and this will help clinicians and those that are doing translational research to better learn how their different therapies are affecting or how they would hope to affect the patient's microbiome. The researchers who generated this database hope that it will speed the development of different pro and prebiotic therapies, as well as be useful in investigating microbiota transplants. This was not written up in any of the news locations that I normally seek out, but it was widely shared on different social media platforms by both science writers, as you see up here, by scientists themselves, and also by those that want to promote gut health and just have an interest in gut health. One could imagine that citizen scientists who get their microbiota sequenced through companies such as Ubiome would then be able to better understand the depth of some of the results by going to these different types of databases. All right, moving on, we're going to next talk about another paper from Nature Communications, and this one is titled Antifungal Tolerance is a Subpopulation Effect Distinct from Resistance and is Associated with Persistent Candidemia. Take-home message from this is that Candida drug tolerance increases difficulty of its treatment. And so we're going to talk about Candida resistance, which is not the same thing as persistence. What we're talking about here are very similar phenomena that allow a microbe to grow or to withstand the presence of a drug. But resistance is generally where you have a large population and maybe a couple of them have a mutation that would allow them to grow in the presence of a drug. Should a patient be treated with a particular drug, only those that have that genetic mutation would continue to not only survive, but also possibly grow in the presence of that drug. And then all of their progeny would have that same genetic mutation. It's a heritable trait. And so should the patient continue to be treated with a drug, it wouldn't affect that particular population. Persistence, or what these authors call tolerance, is a little bit different. It also has to do with a heterogeneous population, but that has to do with the heterogeneity in the metabolic activity, whether different microbes are actively growing or some who are just going to be quiescent, not necessarily in an active reproducing state. And those that are not as actively reproducing or actively replicating can often withstand the presence of a drug because many antifungal drugs, as well as many antibacterial drugs, require that the microbe is actively dividing in order to be to have its killing power. And so persistence, which is a phenomenon that's fairly well studied in bacteria such as Mycobacterium tuberculosis, is known to allow certain members of a population to exist, to continue persisting during drug therapy. And then if the drug is removed, they can start dividing again and repopulate that given niche. And should that drug be re-administered, many of those actively dividing cells will continue to be susceptible. But should there still be some that are quiescently metabolic, then those would allow the seeding of the niche again should the drug be removed. And so this is not a very well studied phenomenon in fungi, and that's what these authors want to look at. What about persistence, which we're going to call tolerance here, in Candida albicans? And so they developed a couple of different assays to study this. One is using a fluorescent azole drug. So fluconazole is an azole drug that must be internalized into the cell to be active. And by using a fluorescent version of the drug, they can look at how much has accumulated inside of cells. And the hypothesis that they had is that the more tolerant cells, because they're not actively dividing, will be able to accumulate more drug inside of themselves and will thus be brighter and can be detected by different types of tests that look at cell fluorescence, such as flow cytometry. And they looked at not only the standard lab strain, as you can see here, SC534, which is a standard lab strain, but they looked at patients' samples, so isolates that came from either patients that had had persistent candidemia, meaning that they would have one bout of disease, be treated with an antifungal, and then that disease would recur over and over again. So it'd be treatment recurrence, or they compared that to patients that had had a single bout of candidemia. So those that had had one infection, been treated with a particular antifungal, and then been able to clear that infection and not return to the disease state. What they found is that the isolates from patients with persistent candidemia that had to be consistently treated or repeatedly treated did have a higher population of these tolerant or very bright fungi. So this suggests that the greater the population of these tolerant microbes, the more likely one is to have a recurrence of disease. Now, how did those tolerant cells achieve the lower drug concentration? What's the mechanism by which they're able to be metabolically quiet and to have that drug inside of their cytoplasm without necessarily affecting the function that it's supposed to inhibit? To test this, they looked at what's called disk diffusion assays. So you can see down here, there's a number of different disk diffusion assays. And if we concentrate on this one, on the middle, on the top, you can see that there's one drug imbued disk that seeps drug into the agar around it, and it diffuses and dilutes as it diffuses away. Oops. And at a certain concentration, there is what's called the zone of inhibition. So that's where you should not see any more cells that are able to grow because the concentration of drug is too great. Now, in these persistent populations, you can see where that zone of inhibition is, but you also see a number of speckles. And in this case, it's almost the same density as the other population, the susceptible population. And these inside of the zone of inhibition are what the authors call the tolerant population that are able to withstand that higher drug concentration without necessarily dying. And so in order to see if there was synergy between the azole drug and any other type of inhibitor, they added inhibitors to various stress responses, things like calmodulin inhibitors, calcineurin inhibitors, and heat shock protein inhibitors that may work in tandem with the fluconazole to render those cells susceptible. And you can see in each of these panels that there is a no adjuvant or no co-treatment panel, wherein there's a whole lot of these tolerant cells that are persisting inside of that zone of inhibition. But with the addition of a number of these inhibitors, they were able to render all of those cells susceptible, suggesting that these different stress response pathways play a role in allowing the cells to become tolerant. And this does, in addition, offers a little bit of insight into what other types of inhibitors may work as co-therapies alongside some of these azole antifungal drugs. And so this was something that was shared on social media. You could see both of these particular users are involved with the study, the senior scientist and one of the authors, who shared a blog from the Nature blog community. That's the Behind the Paper blog. We'll link to that below, along with, of course, all of the original studies. But this is a nice way to get a little bit of insight from the author's point of view about what the experiments were that were done and some of the challenges in those experiments. There's also a commentary that had this very lovely figure illustrating the high and low tolerance and the high and low tolerance within a population and how that can affect treatment and lead to either clinical persistence or infection clearance. And I also wanted to discuss this paper because Candida has been in the news quite a bit recently, both in our sessions that we've talked about, as well as a recent piece about the strange and curious case of the deadly superbug yeast. Now, this is not about Candida albicans, which is the yeast that we've just discussed, but about Candida auris, which is a new, very highly resistant fungal pathogen that was recently talked about at a mycology meeting by mycologist Tom Chiller, who said that this bug is the most difficult we've ever seen. It's much harder to kill. And the hope is that some of these Candida studies on related sister species may eventually be beneficial for coming up with therapies for some of these harder to treat species, such as Candida auris and others such as Candida glabrata. And so you can check those out. We'll link to the blog and to this other Wired story below as well. All right, we're going to move on to our next What's Hot in Microbiology. And here we're going to talk about a paper from The Lancet. This is titled Evaluation of a Mosaic HIV-1 Vaccine in a Multicenter Randomized Double-Blind Placebo-Controlled Phase 1/2a Clinical Trial in rhesus monkeys. So this is a pretty exciting paper. Take-home message is that HIV vaccine elicits strong immune response and enters Phase 2b trial. So HIV, despite the advent of our antiretroviral therapies that are available, continues to be a scourge of humankind. In 2016 alone, there were 1.8 million new HIV infections, bringing the total worldwide to 36.7 million infections. And although many people are able to live for many years using these highly active antiretroviral drugs in combination, there are also many, many more that do not have access to those drugs. And there were, as a result of this, 1 million HIV-related deaths in 2016. And so this has proved to be a very hard to treat disease, and it's also proved to be a very hard to prevent disease. Vaccines, despite so many brilliant minds and lots of funds and many different potential avenues, vaccines have really had limited efficacy. There have only been 4 vaccines that have progressed to Phase 2b. That's where they're tested for their efficacy and not merely for their ability to be immunogenic and safe, which is what we're going to talk about today. I believe one of our previous sessions, we talked about the most recent Phase 2b trial, which occurred in Thailand, and that was the most promising so far, which conferred about 33% protection, but was not deemed high enough to warrant further studies because 33% protection is not high enough to have a positive effect at the population level. And one of the hardest aspects of HIV, one of the aspects of HIV that makes it very difficult to protect against is that there is a wide variety of circulating strains. And so any vaccine must protect against greater diversity than what the circulating strains are that are present, because if it only 1 or 2 or even half of those circulating strains are protected against, it may simply select for those that are not protected, that are not included in that vaccine. And so in this particular trial, we're going to be talking about 393 participants. And these participants were recruited at 12 clinics from around different places in the world, including Thailand, East Africa, South Africa, and the US. And this was so that there was a good genetic diversity of the human population to show that this would be highly effective in many different genetic backgrounds. And these participants were given the AD26 adenovirus-based vaccine at weeks 0 and 12. And so this vaccine has mosaic, meaning different combinations of ONV, GAG, and POL. These are the major antigens of HIV. And then they were followed up with 1 of 3 potential boosters. Some were given no booster, some were given a high-dose booster, and some were given a low-dose booster. There was a parallel study with these same conditions that was conducted in rhesus monkeys, and these rhesus monkeys were also tested for their ability to prevent and protect against HIV infection. So let's look first at the human data. The top here, we're looking at the humoral response, looking at the antibodies that are made in response to the GP140, which is, again, one of those HIV antigens. You can see those different booster groups, either with the high dose, low dose, or no dose, no booster, in a couple different replications here. And you can see that they are showing high response above the baseline. So before the participants were given the vaccine, they had no response. And then there was also, importantly, placebos that were included. These were given 0.9% saline in place of the vaccine. And there was a high titer, meaning a high concentration of antibody in either of the boosters. The boosters did seem to be a little bit higher than the no booster group. And this high titer, this antibody, lasted from week 28 when they were first assayed to week 52. So there was memory, or at least a good response that was longer lasting than simply being half a year. It seemed to reach nearly a year, which is as long as this trial lasted. The researchers also looked at the cellular immunity, looking at the T cells through ELIS spot, looking at the number of responding cells per million peripheral blood circulating cells here. And you can see that there also were high responses compared to the placebo and the baseline effect. And so these robust responses seen in humans were also seen in the monkeys, the rhesus macaques that were tested using that same vaccination scheme. When those monkeys were tested for protection by administration of 6 simian human HIV, it's a SHIV hybrid virus, there was a 67% protection observed, which is better than that 33% that I had mentioned earlier. And it shows a lot of promise for this particular vaccine as it moves into phase 2B clinical trials. So this was a big deal. This is, as I mentioned, only the 5th now vaccine to enter phase 2B clinical trials. This was something that was covered by a number of major news outlets, including the BBC here, and they interviewed one of the senior scientists involved with the study, Dan Baruch, who seemed to couch his enthusiasm here by saying the challenges in the development of an HIV vaccine are unprecedented, and the ability to induce HIV-specific immune responses does not necessarily indicate that a vaccine will protect humans from HIV infection. And I think that a number of these somewhat tepid quotes from Baruch and others involved with the study are mainly due to the fact that the HIV community has been burned so many times before with promising candidate vaccines that have shown robust immune response but not been able to protect against the wide array of those circulating strains. This was shared by many different individuals, many different foundations, communication groups, news reporters, and is something that, should these Microbial Minute sessions continue for another couple years, I'm sure that we will talk about some of the results as they start to come in. Fingers crossed for good results with this particular HIV vaccine. Moving on, we're now going to talk about antibiotic resistance. We've got a number of different aspects of antibiotic resistance to cover, and this is something that we talk about quite often on these Microbial Minute sessions, the problem of conserving the drugs that we have. So the ability to use the right drug at the right time, in the right concentration, for the right duration, is known as stewardship, using the right drugs only when they are necessary and not when they are not, so that we conserve them and make sure that they're useful for as long as possible. And there's a number of different stewardship aspects, right? You can talk about, for example, the consumers should only ask for antibiotics if they have a bacterial infection and understand that a viral infection will not necessarily be treatable by antibiotics. But another potential place for stewardship are the prescribers, the healthcare workers who give out antibiotic prescriptions. And so we have 2 different studies that are looking at whether these antibiotic prescriptions would be good targets for stewardship campaigns. Take-home message is that yes, they would be. So let's first look at a JAMA Internal Medicine article titled Comparison of Antibiotic Prescription in Retail Clinics, Urgent Care Centers, Emergency Departments, and Traditional Ambulatory Care Settings in the United States. This article starts by giving the background that there is 60% of outpatient antibiotic prescriptions that are written in medical offices and ERs. That part is pretty well established. Now, where do the rest of the 40% come from? What this group hypothesizes is that urgent care centers and retail clinics contribute to that 40%. These are growing segments of healthcare providers. They are things such as the Minute Clinic, where you might go at your local pharmacy and get a quick antibiotic prescription should one be warranted. So to look at this, they did a retrospective study looking at data from 2014 involving respiratory diagnoses. So they weren't looking at all antibiotics, but only those for people who had, let's say, a cough or something that was caught in their chest. And they were able to see that indeed urgent care and retail clinics do contribute to that 40%. They are a major part of the remaining antibiotic prescriptions that are written. And then when they looked at the inappropriateness versus the appropriateness of antibiotic prescriptions, meaning whether a prescription for antibiotics was warranted based on the follow-up labs demonstrating that it was a bacterium and a bacterium that was susceptible to a given antibiotic, they found that urgent care centers had the highest rate of antibiotic inappropriate respiratory diagnoses, with about 50% of those prescriptions being unwarranted or given in an inappropriate fashion. And so this will definitely help stewardship campaigns to better target not only to the consumers, but also to the healthcare providers to make sure that any drugs that are prescribed are the appropriate ones. This is not only an issue in urgent care centers, however. Another study in American Society for Microbiology's Antimicrobial Agents and Chemotherapy looked at a similar issue in a VA healthcare system. This is from a paper titled Prevalence of Inappropriate Antibiotic Prescribing in Primary Care Clinics within a Veterans Affairs healthcare system. In this study, the authors looked at a year's worth of antibiotic prescriptions. So this totaled 3,880 acute antibiotic prescriptions from a total of 767 PCPs, or primary care physicians, who dealt with more than 40,000 patients. And they found that there was a median of these physicians to give 84 antibiotic prescriptions per 1,000 patients per year. So that's quite a high percentage of people who are receiving different antibiotic therapies. They then looked at 300 of these prescriptions in depth. And this was only from a select number of the doctors, but they wanted to look at the different types of infections that were treated. They wanted to look at the different antibiotics that were prescribed. And what they found is that 75% of these reviewed prescriptions were inappropriate in some sense, with the biggest culprits being prescriptions for ciprofloxacin and azithromycin. So if we look at the graphs over here on the right-hand side, you can see that they separated this by acute respiratory tract infection, urinary tract infection, and skin and soft tissue infection. And the color coding here is optimally prescribed is green. So you can see this tiny little slice, 11% in both skin and soft tissue infection and the respiratory tract infection, and 29% in the UTIs were optimally prescribed, meaning that it's the right drug given along proper guidelines, and it was given for the right duration, the right concentration for the right duration. But the rest of that, meaning 88, 9% here and almost 3/4 over here, were given in some sort of inappropriate fashion. Now, it could be that an antibiotic was not indicated, which was a large number of those inappropriate antibiotic prescriptions, but it could also be something like it was given for the wrong agent was given or for the wrong duration. And this definitely indicates that prescribers are a great target for some of these stewardship campaigns. And so this was this is something that is not only an issue in the United States, and is not only an issue from prescribers. Just really quick, wanted to point out a Journal of Infection study that also recently came out discussing the issue of over-the-counter antibiotics and how this is also contributing to inappropriate use of antibiotics when the drug is not actually going to be able to treat the given infection, whether it's because the infection is already resistant or because it's from a non-affected microbial type. And this is a big deal, understanding how our drugs are being used, because we want to conserve them and make sure that they're useful for as long as possible. By having stewardship campaigns, the goal is to keep those drugs useful to benefit healthcare and human health and the outcomes of people who go to see the doctor, so that when they do need a drug, it's effective against their particular infection. And this is becoming more important as large pharma is dropping antibiotic development programs. Novartis was the last one who just this last week released a statement in which they said that while the science for these programs is compelling, we have decided to prioritize our resources in other areas where we believe we are better positioned to develop innovative medicines that will have positive impact for patients. And this was commented on by an executive director of a nonprofit that is trying to develop antibiotics, that this Novartis decision is entirely due to the broken business model for antibiotics. And that's because once a new drug is developed, it's going to be conserved and only prescribed very rarely when it's extremely needed. And so the financial incentive is really not there for a lot of these large pharma companies. Pfizer has just absorbed recently another antibiotic development program from, I forget what the company is, but these big pharma is definitely investing in other areas of human health and even other areas of infectious disease, and that's something to be aware of. Now, there's a possibility that smaller companies, as covered in this Stat News piece, may be able to develop some of those antibiotics, those antimicrobial drugs, which are so needed, but that is yet to be seen. Of course, if Big Pharma is leaving this area, it does mean that it is open and available for other companies to move in, and with hope that will be what happens, but I guess we'll just have to wait and see in the coming years. All right, we're going to talk about our last story for the day, and this is a quite simple story. And take-home message is that the FDA has approved a smallpox drug. This is from a press release from the FDA, which the entire title of that press release is that FDA approves the first drug with an indication for treatment of smallpox. Now, why are we talking about smallpox? This is something that is not commonly thought about in infectious disease worlds. It's a drug it's a virus that's highly contagious, but it was eradicated in 1980. And after that, the vaccine stopped being administered in just about every country around the world. This means that most people who are 40 or under, and perhaps even older, depending on when that vaccine campaign ended within a given country, are susceptible to smallpox. And so should smallpox be used as a bioterrorism agent, this would lead to mass chaos. It would lead to a lot of disease, and it would also disrupt society as we tried to deal with this bioterrorism agent. And so to combat this possibility, the FDA approved a new drug called TPOXX under the Fast Track and Priority Review divisions, which TPOXX also received an orphan drug designation. And in their press release, FDA's Commissioner Scott Gottlieb made a statement saying that to address the risk of bioterrorism, Congress has taken steps to enable the development and approval of countermeasures to thwart pathogens that could be employed as weapons. Today's approval provides an important milestone in these efforts. This new treatment affords us an additional option should smallpox ever be used as a bioweapon. So really, this is an approval that is a just-in-case approval, but it's important to have something on the shelf should it become necessary. And so what is TPOXX? TPOXX is formerly known as tecovirimat, and formerly known before that as ST246. It was identified in a high-throughput screen of over 350,000 compounds. So those of you who are working on those high-throughput screens, it can sometimes pan out and develop something that's very useful. This particular drug targets the orthopoxvirus P37 protein, which is a great target since there are no mammalian homologs, and so it has a very good differential score. P37 mediates the formation of enveloped viruses, and so it's important for the virus to be released in its enveloped form. You can see over on the left-hand side a survival curve of non-human primates that were infected with variola virus, the virus that causes smallpox, and those that were given either placebo or treated at days 3 or 4 with this T-pox. And you can see that that led to their survival at much higher rates than those given the placebo. And of course, as mentioned in this AAC article, it would be neither feasible nor ethical to conduct clinical trials to evaluate the efficacy of tecovirimat against smallpox in humans. And this study, along with another showing the safety in one of those phase 1 trials, that, like the one that we just discussed, showed that it is safe in humans and effective in non-human primates, the closest that we can get to a human model without necessarily involving people. And so the hope is that should there be a smallpox event, that this effectivity inside of primates would translate to being effective inside of people. This was something that was also widely covered, both by very mainstream news such as Forbes and Time. It was shared by the New York Times and also shared by a number of people on social media. And in looking up facts about smallpox, I also found this interesting tidbit showing that in the 19th century London, around every 12th person died of smallpox. Really just a fascinating and compelling case for the efficacy of vaccines, which is what has rendered smallpox virtually unknown to today's society. All right, that's going to summarize, or that's going to be the end of today's Microbial Minutes session. I'd like to thank you all for listening. What have we learned about today? We discussed new databases that link the microbiome composition to disease. We discussed candida drug tolerance that increases difficulty of its treatment. We discussed HIV vaccine that elicits strong immune response and is entering phase 2B trial. We covered the antibiotic prescriptions that are a good target for stewardship. And we talked about the FDA approval of a new smallpox drug. If you like the news and you like microbiology, go ahead and subscribe so that you can get a notification the next time we'll be having these every few weekly sessions of Microbial Minutes. I'm Julie Wolf. I'd like to thank you all for your attention, and I will talk with you next time.